I never wear pink ever. So don't like that. I don't like it usually. It's just like I had clothes that were clean and clothes that were dirty and it's clean. So. So we're wearing it today.
That's exactly what I thought you said. Okay, fine. Chill. It's like, I did. Just didn't have it. Where she's at. Obviously, nothing's going on. Well, on the last slide of my presentation, I really thought about making a thing that was like, okay, now that this is done, this is 10 reasons why I think you should read Avatar.
I'm in the third book now.
You are?
Oh,
Oh,
I
righty.
Oh,
Yeah. Are you really a fast reader?
I say this, I get dedicated. I get dedicated and then I get a little obsessive and that's what I do in my free time now.
Uh-huh.
Read that. And I'll go through this period where I will read a lot and then eventually in a couple months, I'll probably be not reading at all. And you are pressed. I just go through phases like that.
It's like giving people to join a cult, but it's like the best cult.
I hear Fourth Wing is pretty good too. I think I'll do that
Started
next.
reading, but I did not have time for some. But right when this class is over, I'm going home and reading. Yeah. Ready. There were some. You have to look at those. I only have the first one, but I wanted to wait to get the third one until we get a paper back. So you can make it. Yeah, they just don't have honest paper back yet. They might. I just don't like. Okay.
This, I don't want to see this. All right, everybody, make sure that you are signed into the Kahoot. We're going to get started here in just a second. Make sure we're signed, or yes, sign into the Kahoot.
Students typically do better on this lab exam or the midterm exam. Yeah.
It's boring. That's the real reason for it.
Yeah.
It's just that information. It's just a lot.
The thing I'm most nervous about is like seeing the tubes, but then there's so many tubes with that same color that it just messes my brain.
Yeah, that's the problem. I think that what Phillips will have is just a lot of colors.
Yeah.
Like what the colors mean and what situations, stuff like that. So, yeah, that tends to be the hardest thing for a lot of people.
There's like questions with them, not just like, what is this?
Yes. So there will be some that'll be limited.
Okay.
Yeah. So there'll be some where I will give you, maybe not a plate, but I might give you a two. This.
Yeah.
What is it? Right?
Yeah.
So, yeah.
That's.
You get what I'm saying? Yeah. So there will be some. There will be plenty of questions like that where I will ask, what's the pH indicator in this media? I'm not going to tell you what the media is. Or I'm going to say, did this organism ferment lactose? I'm not going to tell you, you know, what color it should be if it does ferment or whatever. So you need to know that stuff.
That's so good.
So we'll go over some of that stuff today. Try to see if I can get. take a picture of the board. If I write anything on the board today, for those of you who are joining via Zoom, if I write anything on the board today, I will take a picture of it. You might be able to see it from where you're at. Hold on, let me see if I can work the camera, the overhead camera. Fantasy, ivory, dark, really as well. I don't like it. Kind of see it. No. Make it a little bit higher. So I will take a picture of anything I write on the board today. Okay, so make sure we're signed in, double check, double check. We're gonna go ahead and get started with the Kahoot. We've got 2, 4, 6, 8 in here, and we've got 2 signed in online. Yes, we are recording this, and yes, I will post the recording afterward for you guys to review. Let's go ahead and get started. Alrighty. I will show you real tubes when I have real tubes, okay? So first things first, we're gonna go over, basically the lab final is gonna cover all of our media basics, what broths are, auger, isolation methods, microscope parts and how they function. I would know which lens creates the real image and which lens creates the virtual image. I would know that for the final. Possibly microscope magnification calculation should be an easy one. Nobody should be worried about that if that's on there. Lab safety, there will absolutely be a lab safety question on the test. You're welcome, by the way, for that, because it's not going to be a difficult one. And then all of our biochemical tests, which is really the focus of the exam. Most of the questions will be that. All right, so we'll have basically a station at each spot where people sit. So it says 24 to 26, it's going to be 24. Actually 23. It'll be 23 because that station back by the door back there where question 1 was last time, oh, that's going to be 1 spot. So we don't have to keep us having, remember how last time we'd be like, you have to get past the person that was at the next spot. We're not going to do that this time. It's going to be 1 spot there. And so I think there's 3 questions there. But if you have a station that has 3 questions, then they're typically easier questions. But if you have a station that has 1 question, for example, questions 49 and 50, those are separate stations. So obviously those are going to take a little bit more time. Again, same thing as last time, it's only 50 questions. It's all multiple choice, no like multi-choice questions where you have to select all that apply or anything, but there might be a choice that is all that they apply. You know what I mean? So it'll be just the same format, multiple choice, 50 questions, 1 minute at each station, and then you have more than enough time afterwards to go back and visit any stations you want. There's no real time limit on the exam. Technically, I'm supposed to give you guys a time limit of a class period, but I don't care. So it's like, you can still show me that you understand the material. I don't really care how long you take to do it. That's what's more important to me, is that you show me you understand it. So don't worry about the timing. Yes, we're going to do the 1 minute rotations, but remember, you can go back to any station. There will be plenty of time, even if there's a line at the station, there will be plenty of time. So don't stress about the time part, all right? Same as last time, keep your laptop in your little cubicle thing. So keep that in mind as well, okay? So I'm just got those out for now, and welcome, just move them around as you need for now, but it's just gonna save me time to set up the exam right after this. So it's just a lot of work to set it up, so I'm just getting stuff ready for it right now. Yes, so we know what to expect, but just to review, remember to wear your lab coats and your closed-toed shoes. Same rule as last time. If you have to borrow a lab coat, it'll cost you 2.5 points. That is a massive lie. If I don't have any lab coats, then that would cost you 5. And if you don't have closed-toed shoes, then I'm going to send you to the testing center, and you'll have to take it in there, and you'll get a 5-point deduction as well. So keep that in mind. Don't mess with it. Closed-toed shoes and lab coats tomorrow. Okay. All right, this counts as closed-toed shoe as far as I'm concerned. If there was broken glass on the floor, which we've seen that happens, right? In this semester, you guys have been better about it than any other semester, but it happens. We sometimes drop a tube or whatever, and the little shards or whatever get moved around the floor. That's the whole reason why we have the closed-toed shoes more than anything. So we're going to start off with our culturing basics. This is going to be our inoculation and general growth stuff. What is most commonly used to inoculate cultures? Again, this is pretty similar to like our last, how we start off. We'll start off easy, and then we'll get into more details that are more specific. All right, so this is the inoculation loop. Most of us got this one. Most of the time, most commonly, we're going to be using that inoculation loop. So we can incinerate and everything like that. Keeping everything as sterile as possible. We do sometimes use the swabs, but more commonly it's going to be the loop. In which quadrant of a streak plate would you expect to find the most single isolated colonies? This wasn't a literal, this was literally on the test for the lab midterm, and it will be on the final as well. You're welcome. Okay, so that's quadrant 4. That's where most of the cells get the most spread out. So we get the isolated colonies that aren't touching anybody else. We can start a pure culture from that. Does everybody remember what a pure culture is? A pure culture is one species of bacteria, only ever one. A mixed culture, if it's relevant, I'll tell you on the test, like I did for the midterm. If it's a mixed culture, I'll tell you. If it's pure culture, if that's important, then I will tell you. Otherwise, assume that it's a pure culture, because I'll always tell you if it's mixed. So remember aseptic technique and lab safety stuff. Where do we put broken glass? The sharps container, right? And that sort of stuff. Types of media, know that agar makes things solid, right? The broth is the liquid one. Different ways to inoculate the media. The needle is what we use for our stabbing our butts, but pretty much everything else uses the loop. But you might swish it in the broth or you might streak the surface, and that's pretty much how it'll go. One of the things that you do have to know, and we're going to get into it in some of our questions with our media next. If I give you this tube, all right, so this tube, and if you guys can see it on the camera, it's green slant. All right, I give you this green slant. How did we inoculate this media? So this was streak the slant. That's it. We didn't stab the butt of this one, nothing. It was just streak the slant. There will be questions about that on the test. Okay, be prepared for that. How do we inoculate it? So then you better know, how did we inoculate this one? Right, this one was the butt and the streak, that's the surface, right? That was this one. All right, move on. Getting there, getting there. All right, let's do the stains real quick. Let's review our stains. Any stain that we've done up to now, really the ones you need to know, there were 4 differential stains that we had to know. The gram stain, the acid fast stain, the capsule stain, and the endospore stain. Right. Okay, so let's see what you guys recall from that stuff and review them as we go. What is the first step in the gram stain procedure? All right, so that's crystal violet. Crystal violet is our primary stain. So obviously first step would be to heat fix the slide, right? Make your heat fix thing. But as far as the staining process goes itself, crystal violet is our primary stain. The crystal violet itself gets caught up in the thick peptidoglycan wall of the gram positives. Next step would be the iodine, which is our mordant, which might be the next question, I don't know. But hey, the mordant iodine fixes the crystal violet in place. The step after that is the alcohol. Alcohol acts as the decolorizer. It removes any crystal violet that didn't get stuck in place by the iodine. And then safranin is our counter stain for this one. Not secondary stain, it is the counter stain. All right, next question. What color do properly stained gram-negative bacteria stain in the gram stain procedure? On the exam, I will never, for a gram stain situation, I will never put pink and red. Never. Because I like to just remember it as red, and that's easy for me, so I'm trying to make it easy for you guys, okay? So safranin itself is a red stain, technically. Do the cells look pinkish? I mean, yeah, they do, right? So these flies are really annoying. Anyway, so red. If you're gram negative, you're red. If you're gram positive, you are purple. And it should be pretty obvious, but if you guys can't tell what color it is in the microscope, just ask me, is it purple? Or I think this is purple. And I can come over there and at least tell you it's purple, okay? I've had students come through my class that were colorblind that literally couldn't see the difference between colors. And so I had to tell them every test, like what color a thing was. So they had to know what the color meant. That was on them. So if you guys need clarification on what color it is, that is free. You get that. All right, what is this staining procedure? Better bet it's going to be on the exam. This is the endospore stain. This is the pure culture, and you see red, and you see these round things, red rod, and there's these weird round things all around, and some of them are kind of greenish tint to them, and that's the endospore stain always, especially if it's a pure culture. That's not a mixture of different bacteria. That's one species of bacteria. And those little round things are the endospores, and the inside of the endospore stain green because of the malachite green and the steam that forced that stain in there. It's the endospore stain. Next question, which of the following bacteria would be acid-fast positive? Be sure you read your choices. Be sure you think through it. Don't let this trip you up on the exam, right? So, all right, so the acid-fast stain, what happens is we use the primary stain is carbofuscin, which is lipid soluble. That carbofuscin lipid soluble stain is what's going to stain mycolic acid wax, which is a lipid. Organisms that have mycolic acid wax on their surface are Mycobacterium species. Remember mycoplasma, the important thing with that guy is that the plasma, the spilled cup, it doesn't maintain its shape. That's the ones that have no cell wall. They have no cell wall, they can't have mycolic acid on their wall. So mycolic acid is present on the wall of gram-positive bacteria called Mycobacterium. Okay, good job. Next question. After the slide has been heat fixed, when do we use heat again in the acid-fast stain procedure? Acid-fast. We did have this question in our lab midterm review. Let's see if we can recall it. All right, so this answer for this one was no. This is because we aren't using heat again. What stain procedure does use heat? The endospore stain. So after heat fixing the endospore stain, we'll use the steam, the heat to fix the malachite green in. But for acid fast, all we did was we made our heat fix stain. We added our heat fix smear. We added our carbofuscin primary stain for 5 minutes, rinsed it with acid alcohol, and then counterstained with methylene blue. That's it for the acid fast stain. That was it. All righty, next question. What color is a negative acid fast stain? If you are not acid fast positive, you don't have mycolic acid. What color do you stain with the acid fast staining procedure? So I've told you that our primary stain is carbofuscin, that it binds to mycolic acid. Carbofuscin is fuchsia. But what do we use as our counter stain to stain if you're negative? We use methylene blue. So if you are negative for mycolic acid, you will stain blue. Beautiful. You guys are doing great. What is the name of the primary stain used in the capsule staining procedure? So this is an image of the capsule stain, what it might look like afterward. So look at this. We're going to come back to it here in just a second. What is our primary stain? Yeah, so our primary stain is Congo red. So when we look at this picture here for our capsule stain, we see that the background is all purple. And we see that there's little like pinkish red structures in the middle of the white. So the white is the capsules, right? The capsules don't stain, they create that halo. So what are those pinkish red structures? I mean, basic. They're just cells, they're bacterial cells. The pinkish red things in this image are the cells themselves. The purple is not the cells. The purple is just the background. It's the space in between, okay? The white is the capsules. So the pinkish red is the cells. We would say that the pinkish red is cells that have capsules in this, right? So quite obviously they have capsules in this image. So you can see the white around the pinkish red structures. Are we tracking? We can analyze that. So Congo red, it stains the background. I think this is the next question again. Oh, no, it's not. Okay. All right. In which staining procedure did we use steam? I thought it was going to be the right, so that's the endospore stain, which we went over just a little bit ago. We will use the heat steam to force the malachite green into the basic coating of our endospores. So real quick, before we go on to that, let me see if I can go back. This capsule stain one. If we use Congo red as our primary stain, what type of stain is Congo red? It is a negative stain. Remember, negative stains stain the background. So I wanted to mention that again. Remember, negative stains stain the background. Positive stains stain the cells. We already did. All right, good. All right, so know the procedures, not the times. I'm not going to ask you for how long each step has to go. I'm not going to ask you much about the deionized water either. So definitely know your stain steps, know what the positive looks like, what the negative looks like, and why the positive looks that way, and why the negative looks that way for that staining procedure. Know what each step does, because every step has a purpose, and know what they are for each of those stains. So we know what to expect for the types of questions. The types of questions for the final exam will be very similar types of questions, but they may not be the exact same ones this time. So be aware of that, but it should be the same types of questions. Nothing out of the blue with those, okay? All right. This is just so let's get into our differential and selective media. This was our EMB, blood auger, staph media, all of that stuff. All right, so which type of media is this? Yes, questions will be like this on the test, some of them. Which media is this one? So just so we can be clear here, this is metallic green growth on here. This is what the plate will look like on the test. You'll get actual plate. This is what the plate will look like on the test. You guys can see it looking on the camera. This is ENB, ENB. What is the selective agent in this media? In EMB, what is the selective agent? So just to remind you guys about selective and differential. Selective media means some things grow and some things don't. Differential media means you can tell the difference between things that do grow. It's usually color changes, right? The selective agent in EMB is going to be the EMB itself, eosin methylene blue. Those are molecules that basically inhibit the growth of gram positives to favor the growth of gram negatives. Okay, so EMB has EMB as its selective agent. With EMB, is it differential also? Is it differential? Yes. So remember, if the growth's on here, and this is the plate where we have metallic green, right? So we can say because it's metallic green, we are an aggressive lactose user or catabolizer. Breakdown. Remember, cats break things down, destroy. So we are aggressive lactose catabolism. If it was just regular lactose catabolism, what color would it be? Purplish pink. And if it didn't use lactose, what color would it be? No color change. Okay. All right. Beautiful. My hands are going to get gross today. I can already. All right, which media is this one? It looks like this. This is the plate. This is one where I will usually on the exam tell you what the media is because it looks like some other media that we use. So this one I will usually tell you what this media is. This is McConkey auger. Is it selective or differential? Both. Excellent. So it's both, right? I think there'll be some questions coming up. McConkey auger is both selective and differential. All right. Oh, okay. That's the next question. It should be both selective and differential. I'm using a lot of hand sanitizer there. All right, so it should be both. Excellent. Easy enough. So it's selective. I think we'll just do it. I don't care if it's the next question, but we'll review it now. What's the selective agent in McConkey agar? Anybody know? Bile salts. Bile salts. All right, bile salts. So this favors the growth of gram-negative enterics. It also has crystal violet in it. So crystal violet gets stuck in the gram-positive wall, just like it does in the stain, and inhibits the gram-positives from growing well. So the gram-positives don't grow well typically on this, right? The gram-negatives typically grow better, and the bile salts favor things that can survive the gut environment. How is it differential? What does it look for the use of? Lactose, right? And it's the same kind of as the EMB, but without the metallic green, right? So we have pinkish color if you use lactose, and you'd no change if you didn't. It's probably the next question, but we reviewed it. Okay, no, it's not good. What media is this one? This is the plate that you'll see on the test. Or it might be a newer one. I might reculture it just so that it's a little real life plate. All right, so this is mannitol salt agar. And if I put it on the test, it'll have the yellow, okay? So I feel like that's the defining characteristic with this one, okay? So that reddish color to that yellow shift. This is mannitol salt agar. Is it selective or differential? Both. Excellent. So what makes it selective? Salt, right? Gram positives with their thick wall survive better on salt than gram negatives typically. And is it, so we know it's differential. What does it look for the use of with its color change? Mannitol, right? Breaking down mannitol, fermenting it. If it makes acid, it turns the pH indicator yellow. What is the pH indicator used in mannitol salt agar? pH indicator. This media. All right, so this is where I'm going to draw on the board a little bit to help you guys a little bit out with our pH indicators. For people who are online, I will take a picture of this and post it afterwards so you guys can see it. But basically, for the pH scale, we know pH neutral is 7. So this is neutral. This is pH scale. 7 is neutral. 14 is alkaline. Or basic, whatever you want to call it. They're the same thing, okay? And then 0 is towards acid. The lower pH, the more acid. Another way to look at it, the lower the pH, the higher the hydrogen ion concentration, which will be on your lecture exam, all right? Just to recall that stuff. The hydrogen ion concentration stuff will be on the lecture exam, not on the lab stuff. I just want to tie it all in. Okay, this is our pH scale. We had 3 different pH indicators in our media, just 3. That's it, just 3. Okay, so we had phenol red. We had litmus in order of their introduction to us. And we had bromothymol blue. I'm making a chart for those of you guys who are wondering what's happening on the. Okay, so I've listed all 3 of our pH indicators. What color is phenol red at neutral? Reddish coral color, right? What was litmus at neutral? Lavender, right? And what was bromothymol blue at neutral? It was green, right? Yes, I know. Okay. So, phenol red. When we are acid, what color do we use? Yellow, just like we just saw, yellow. Usually happens from acid production from fermentation, most of the time. Fermentation usually is what makes the acid. All right, for phenol red, let's do all phenol red, yeah. For phenol red, what's the alkaline look like? We did have this one in some of our tests. Pink. It's pink. What test relied on pink for positive with the phenol red? Urea. Remember how urea turned bright pink? Okay, so we'll come to it in a little bit, but we'll get there. Okay, so pink. All right. Litmus. So remember with our phenol red, most of our media turned yellow from fermenting a carbohydrate. Most of our media turned pink from breaking something down to make ammonia, a nitrogen source of some kind got broken down to make ammonia. So usually that's what's happening here. So I'll even make a little note of that. Phenol red usually turns yellow. You know yellow is acid. From fermenting. From acid from fermenting. The pink, the alkaline, is usually from ammonia from our nitrogen source. All right. Litmus. We got some weird stuff going on with it, so I'm actually going to divide it up a little bit. Litmus. When we have acid present, what color does it turn? Just regular, just acid. It turns pink. Actually, put it up top. Pink. This is from fermenting lactose. Again, fermenting makes acid. If we really made a lot of acid and it started changing the litmus itself, we called that reduction, right? So that was white. Where the litmus itself got reduced from excessive acid. So I'll put that even further out on the acid scale. All right. If we're making alkaline, just at first, our first stage of alkaline production from breaking down the nitrogen source on our litmus, what color does it turn? Blue. Blue. Blue happens with litmus. Same thing as up here. Nitrogen source breakdown. I just put nitrogen source. Nitrogen breakdown. All right. If we really break down the proteins, not just breaking down just as a nitrogen source, we continue degrading the proteins even further, which is an extreme end on that alkaline scale, then we get to digestion, which is clearing up. So if it goes clear or translucent, then we have digestion of the protein. In the white area, you might also have coagulation, which is what it sounds like, curdling of the milk. That happens in the white end, just to remind you guys. You see chunks in there, usually there's white also. Some things I want to show you with the litmus milk, because I forgot to grab it. Litmus milk, uninoculated, lavender, okay? I'll come show you guys the camera in a little bit. Litmus milk, actually, let me take it up here so I can show that. Litmus milk, lavender, when it's neutral, put that in the middle. If we make acid from fermenting, it'll be pinker. Pinker. And I'm going to hold it up next to the purple so you can see that it is pinker. You guys see it? All right. These will be the tubes you'll see on the test. So I want to be sure I'm going over them. So we've got in the back. Line up with this. Lavender, pink, acid. All right, if we keep doing that more, we can see there is some pink and there is some clear up here. But why is there clear up here? Because we coagulated. You see how chunky it got in here? So this is a reduction. We have reduction here. We have acid here. This does not count as digestion, though. This is not true digestion because we know that because the liquids just separate, it's just a whey, you know, curds and whey. Guys, we haven't actually broken down the protein. It's just separating from that coagulation. You can see the chunks in there. They're quite obviously chunky. All right, so this is that chunky reduction and acid. On the test, I will often ask, of the following choices, what happens to the litmus milk? So I might not even have acid or reduction on there. I might just have coagulation as a choice. Or I might not have coagulation or acid. I might just have reduction as a choice. But we know it's not alkaline, right? We know it's not digestion because digestion happens whenever we clear up the whole thing. What does digestion look like? So we've got purple, but in the bottom it's clearing up. So digestion. The bottom of it's turning like plasma. It's at the extreme end, but that alkaline will just be blue. So you guys can see the difference there. Blue, alkaline. We've got all of our litmus milks there lined up here according to our pH. Neutral, more acid, more alkaline. All right, so walk that around so you guys can see. Neutral, more acid, more alkaline. We go with litmus milk. I'll show that to you guys here on the camera. Those of you guys watching the recording or watching in person. More acid, more alkaline, neutral. All right, so that's litmus. So that's our hardest one just because there's a lot going on with it, right? I'm back over here. I have, there it is. All right, we didn't have to worry about acid being produced with our bromothymol blue test, did we? Just FYI, if we did have to worry about it, and we don't, I'm not going to test you on it. So I'll write it. I don't know, I'll write it, but whatever. It would be yellow from fermentation. But we didn't need to know it. There's a reason I'm bringing it up. I'll come back to it in a second, okay? But we know bromothymol blue is green when it's neutral. And then what color does it turn when it's alkaline? Blue, right. Blue, again, why are we going alkaline? Because of nitrogen source breakdown. Making ammonia. So generally speaking, if you're going alkaline, you're making ammonia. Generally speaking, if you're going acid, you're fermenting a sugar. All right, we track it. Something I want you guys to notice over here. For all of our colors, for all of them, the more blue you are, the more alkaline you are. Tell me that pink isn't bluer than yellow. It is. The more yellow you are, the more acid you are. Even with litmus milk, we went up to blue, lavender, to pink. Pink is yellower than blue on our color wheel, right? So that's how I remember it. The more yellow you're going, and even white, yellower than pink to me. I don't know. Maybe that doesn't compute to you guys, but to me it does. Yellow to blue. You got to break them down and simplify them. That's how I remember them. That way, you've only got 3 pH indicators. You've got one general color scale going on. If you can remember what the neutral colors are, start from there. All right. All right. So, tests that are related to these. Phenol red. We had mannitol salts. We had carbohydrate fermentation. The one with the Durham tube, right? We had urea. And we had TSIA. Am I missing anything? That was it, right? For the litmus milk, what test used litmus as the, well, I gave it away, I guess, litmus milk. The only one. For that pH indicator. And then bromothymol blue, there was just two of them, right? Simmons citrate. Which was the slant. Simmons citrate was the slant. Right? And then malonate broth. If I could lift you guys up and carry you over here, I will, but I can't. So I will take a picture of it and post it. You guys are welcome to take pictures of it too, if you want to. But I am going to take a picture and post it, and everybody will take it.
Are the MRVP tests different than these ones then?
You tell me.
What, with the MRVP tests, they don't count on our active scale test?
They're not pH indicators, really. So there is a chemical reaction there. It doesn't depend so much on the pH. So with MRVP, it doesn't depend so much on the pH as it depends on the actual literal chemical compounds there. So there's an actual reaction going on with the chemical compounds there that aren't really pH dependent, but they do depend on the acids that are there, but it's just not pH dependent. Right. So MRVP, not a pH dependent test. Neither of them are, not pH dependent. All right. They are specific chemical dependent. All right. We good with the pH stuff? Because there are going to be a lot of questions about that coming up, and you are welcome to refer to the board for your Kahoot. But when you guys are taking the test, maybe have a version of this that you write for your scratch paper that has all that stuff on it, so you can refer to it, and it should make it a lot easier. So I will post that picture on when I get done with this, along with the video and the AI summary for this, for those of you guys watching online. All right. Good. All right, so know which media is selective versus differential versus both versus neither. One thing that we didn't cover on this particular review was not it. It didn't refrigerate very well. Was the blood auger. I'll have to make a new plate of it. Blood auger. So remember that blood auger was not selective, but it was differential. So if you grew on blood auger, you had some kind of hemolysis, right? And so with blood auger, and mine did not refrigerate well, so I'm gonna have to culture a new one for the exam, which is fine. But with blood auger, if you grew on it and you didn't change the media, then we would consider that gamma hemolysis. If you grew on it and the media turned greenish or darker or something, then that is alpha hemolysis. But if you grew on it and you cleared an area around it, if you hold up to the light and you can see through it better, that is beta hemolysis, which is complete hemolysis. So know those differences. There will be a plate on the exam. You'll probably have to lift it up to the light to see what kind of hemolysis happened, okay? Or if there was any hemolysis. All right, I was really banking on that refrigerating and it didn't, so whatever. All right, biochemical test. All of the rest of the tests that we did, this part. So which test is this one? This is what it will look like on the test. These are the actual tubes for the test. And I will usually, depending on what the question is, I might just say, what test is this? I might tell you which one is inoculated and which one is uninoculated. So obviously this is uninoculated and this one is inoculated. And you can see that there's kind of two different colors going on in here. So you can pretty much tell which one is the positive tube on this one, right? This one's pretty straightforward. These will be the exact tubes on your test. Okay, exact tubes on your test. All right, so this is citrate, Simmons citrate blank. Bromothymol blue is our pH indicator, which might be the next question, but we've already covered it. That's okay. Which of those two tubes is positive? Another common type of question on the test. Up here, which of these tubes is positive? For this one, this would be your A, and this would be your B. Same setup. This would be your A, this would be your B. All right, so obviously B, it's got the blue. That's our pH change. So if we were asking a question about this tube, it would be, did this organism in tube B, was it able to use citrate as its sole carbon source? The answer would be yes for this one. It turned it blue. If it used citrate, it was able to break down the nitrogen source. It turned it alkaline. That's what's really happening in the tube. Don't just know what positive and negative looks like. Know what's happening in the actual test. So what's happening to that pH though in the tube? What's happening to the pH? The pH went up. Alkaline means your pH goes up towards 14. Excellent. Let's just got that one. Keep your pH scale straight. Know what going up and going down means. So how do we inoculate this one? Surface streak on the slant. How do we read it? We look for that color change. Blue means alkaline. That means positive for citrate use. What colors are produced? Blue. It starts green and it turns blue if it's positive. And what do the colors mean? If it turned blue, that means we made an alkaline product, which is ammonia in this case, from breaking down our nitrogen source. The pH indicator is bromothymol blue. Next question, which test is this one? We see in the picture, there is a little Durham tube. This is one where I almost always give you an uninoculated and an inoculated. Pay attention, it's got a Durham tube. We've only got 2 tests with Durham tubes. And this is what the uninoculated tubes for both of those tests look like. Tell me you can't tell the difference, right? Carbohydrate fermentation, also known as phenol red carbohydrate fermentation broth. I might not give you the phenol red though, because you got to know that it's phenol red in there, right? But the other one is nitrogen broth, and it's very clear. It's almost a clear color. Know them from sight. All right. So if it is yellow, like what we said, it's the yellowish orange in that picture there. If it's a yellowish orange like that, we know it's not the nitrogen broth because the nitrogen's almost clear. This is what our uninoculated tube looks like for our carbohydrate fermentation media. Right. Carbohydrate fermentation media. Next. Know them from sight. What is the name of that upside down tube? I said it a second ago. Can you recall it? Upside down tube in this guy. Same upside down tube in the nitrate reduction media. This is the Durham tube. It collects carbon dioxide made from fermentation, or in the case of the nitrate reduction, it collects N2 gas from reducing nitrate in some cases. All right, collects gas from respiration processes. All right, what are the 2 products of fermentation that we can detect from this assay? If you fermented, what product can that assay detect? I'll show you a tube with both of them after you guys answer. Sure you read the question, think through the answers. Right, so for the product of fermentation, so we'll test for fermentation of sugars, sure. This is a tube that has both of those products. We have made acid, which has turned our phenol red yellow. We can detect that, right? We can also detect gas production. In this case, it's CO2. This is one of the tubes that'll be on your test when we're walking around so you guys can see. This will be the tube. I don't mean one like it. I mean, these tubes that I'm holding up today are the tubes that you'll see on the test. The only difference will be the blood odor because it didn't keep in the fridge. All right, so we can see here, we can see the gas that's trapped in the Durham tube there, right? Gas, which is CO2 from fermentation, and acid, which is usually lactic acid made from fermentation of whatever the sugar is. I'm not going to give you a tube that looks like this and ask you what sugar did it ferment. I might tell you that this is a glucose carbohydrate fermentation tube, and then ask you, about glucose fermentation. But I don't remember that these tubes only test one sugar fermentation at a time. All right. I'll show you guys on the camera so that you guys aren't missing out. You can see it turned yellow and that there's air in that Durham tube there. That's the carbon dioxide from fermentation and the acid from the fermentation. All right. Next. Which test is this one? I don't have one out here, so you have to go by the pictures here. It is a butt, and you will be able to lift up these tubes. So the far left, that's what it would look like uninoculated, basically. Pop one up here. I have to inoculate these the day before. No. Blood auger and right. All right. So that test is the SIM. We know it was the SIM because we had a tube that had a little bit of red at the top, and we had a tube that had black precipitate, which are some possible results from the SIM. Yes, you'll be able to pick up the tubes during the lab exam. Please don't just tip every single one of them upside down like that, though, okay? Only tip them if you need to. And don't tip them all the way, just tip them enough, okay? All right. So that's the SIM. What does the I stand for in SIM? This is uninoculated SIM. Uninoculated. These are some possible results that we might see. All right, so I stands for indole. Indole is a product of tryptophan catabolism. Breaking down tryptophan makes indole. We use a special reagent to check for indole, which I think is the next question. How is indole detected in the SIM media? We add a reagent. Which reagent detects indole? All right, so for indole, we add Kovacs reagents. For FeCl3, that is used in the phenylalanine deaminase assay. Peroxide is used for the catalase test, and zinc is used in nitrate reduction test. The Kovacs reagent detects indole in the SIM, and it creates that layer at the top, which is where we saw that red band. All right, how do you detect H2S, which is a product of sulfur reduction in SIM? All right, this is going to be an observation of a black precipitate. The reagent in SIM that allows us to see the black precipitate is going to be an iron-based reagent, just like the same reagent that is present in TSIA. Hint, hint. Okay, so it's an iron-based reagent, reacts with H2S, which is a product of reducing sulfur. Remember, reduced means gain electrons. In this case, electrons come with the hydrogen. Just like whenever we reduced oxygen to make H2O in aerobic respiration, H2O, the H's came with the electrons. So that's how oxygen gained its electrons. So it's the same thing here. Instead of making H2O, they make H2S. It's an anaerobic respiration process. All righty, beautiful. How is motility detected in the SIM test? I might be able to find it too. How's that? But I don't have it with me. What would it look like if an organism was modal versus non-modal? Test might not be as obvious as this one because I have to inoculate them the day before because because the media doesn't last with the reagent on it, but we'll have one that will be clear and one that's slightly cloudy. The cloudy one is the modal one. Clear line, cloudy. And you'll have to tell me which one is modal. Clear line, cloudy around it. You have cloudy up there? Clear line, cloudy. Show you guys, clear line, cloudy. The cloudy one is the modal one. Everybody good on that? I'll have them with each other. You will have 3 tubes, uninoculated, one that's modal and one that's non-modal, and you'll have to tell me which one is the modal one. Okay? So here's what it looks like next to each other. Clear around the line, cloudy. The cloudy one will be the modal one. The clear around the line is the non-modal. It won't be these exact tubes because these are a little bit gunky at the top. So to add the indole and have it work properly, it has to be a non-modal, but it'll be very similar. All right, moving on. Growth away from the stab line, cloudiness is growth away from the stab line. All right, next question for sim, maybe? Oh, we don't sim. All right, that covers it. How do we inoculate the sim? Stab the butt, needle into the butt, right? Needle, butt, stab. All right, interpretation of results. For sulfur, positive black precipitate, negative, no change as far as black precipitate goes. For indole detection, add Kovacs reagent. If it turns red, you are positive for indole, which is tryptophan catabolism. Okay. If it stays golden brown, no change, then that is negative. The reagent that we use is the Kovacs reagent. What is the pH indicator in sim media? None. No pH indicator in sim. Don't fall for the trick. Okay. Good. All right, which test is this one? I don't know. I'll memorize. Okay. We just did this one a second ago. This one, uninoculated. This is litmus milk. Pretty straightforward. I feel like nobody's going to get that one wrong as far as what kind of test it is. And what the pH, I hope you guys don't get what's the pH indicator. Hopefully you know that it's litmus. Okay. With litmus, what color is associated with an increased pH? Increased pH. pH value going up. That's going to be blue. Excellent. It has to go through blue to get to the digestion. Beautiful. In litmus milk, white color represents litmus milk, white. If you have white, especially in the bottom, and there's a lot of it, what does it represent? On the board, for those of you guys that can see the board, this is going to be reduction. Reduction. If you have chunks, that's coagulation. But the white itself doesn't mean you'll have chunks. You can be liquid with white. But if you are also chunky, if I tip it hard enough, you'll see that it stays almost solid in there. Please don't tip it this far for the exam. Okay, you can see that it's chunky just from tipping it a little bit. Just don't want you guys to spill it. All right. All right, know our colors. We went over this earlier, so I'm not going to do them again. Know what each of the colors means, what reduction and digestion look like. What is happening with digestion is we're breaking down the protein completely. So that's why it becomes transparent. The protein that makes your milk, basically, what is the word? Opposite translation. Opaque. What makes your milk opaque and white? Those proteins are getting broken down. How is gas detected? We will not see gas on the test, but if we had to detect gas, it would be pockets of air in the actual coagulated mass. It'd be air pockets, like bubbles stuffed in the coagulation. We're not going to see it on the test though, so don't worry about it. What is the sugar in milk? Lactose. We know this, yes. Don't forget it. So when we talk about fermentation, when we talk about acid production in the litmus, we're talking about fermenting lactose. All right, which test is this one? one test. Some of them are treated with a reagent, and some of them aren't. I don't have one for this one either. I have to inoculate it the day before as well. What the uninoculated looks like. The plates on the left were they had been inoculated, but they hadn't been treated with the reagent. This is starch hydrolysis. And I'll tell you what the test is. Okay. Might not tell you the reagent, but I'll be like, this is the starch hydrolysis test. And I might give you a plate that has to take the plate. Here's the plate. So I'm gonna give you a plate that's been inoculated on one side and then on the other side. And then I will put the reagent, which will be in the questions in a minute, right? On there to turn the starch a color, which is that dark color. And then I'll ask you a question, which organism, A or B, hydrolyzed starch? That could be a question on the test. Maybe something like that. So I'll usually tell you more information about it. And I'm going to make you look at a plate that looks like this and then tell me that it's starch hydrolysis. No. That's wild. So that's starch hydrolysis. All righty. So which organism pictured is positive for starch hydrolysis? Two organisms inoculated in a line, one on the left and one on the right. One of them hydrolyzed starch. That'll be the one on the left. We know this because of that halo around it, that glow around it. This is where the starch got broken down, right? And what reagent did we add? That might be the next question. What reagent did we add to do that? Iodine. Excellent. Iodine reacts with starch, turning it that dark purple color. A lot of you use that one. Okay, there we go. That is the next question. A lot of you use that on your lab reports. Which, by the way, you guys did amazing on it. Really don't want to belittle that at all. You guys are great with your reports and with your presentation. Stellar. And I mean, maybe you didn't feel like you had to do much to do a good grade on it, but I'm telling you, in past semesters, you guys put them to shame. So I'm just saying, the effort and the attention to detail and the care that you guys had for it you should be proud of it. So, all right, so that's starch hydrolysis. We use iodine to detect the starch. Where there isn't starch present, starch has been broken down there, the iodine doesn't react. That's where we get that halo from. All right, which test is this one? Next test has this one here. We showed it earlier. Little Durham tube action in here. Clearish color, all slightly off clear, and has a Durham tube. Only two tests with Durham tubes. Saw the other one earlier, the phenol red carbohydrate fermentation test. This is nitrate reduction broth. Nitrate reduction broth. All right, next question for it. What do reagents A and B detect in nitrate reduction? A and B. You add A and B to detect what? A specific chemical compound. Which specific chemical compound do A and B detect? All right, nitrite. I knew it would be between those two. We all knew it would be between those two. Nitrite. It's the nitrate reduction broth. A and B detect nitrite. The other reagent that we add, if you're negative with A and B, then we add zinc. And zinc comes late and tests for nitrate. Okay? So A and B detect nitrite. The only things we ever detect in this test is either nitrite or nitrate. So nitrite first, and then only test for nitrate if you need to. So what does zinc detect? All right, so test for nitrate. If I tell you on the test, this tube is uninoculated. This tube, I added A and B only. I did not add anything else. is this tube positive, this tube here, the tube B, uninoculated, added uninoculated and added A and B only. Inoculated and added A and B only. So this red tube, if all I added was A and B and it's red, yeah, question.
Did you upload your drawing that you about
I mean, it's the same drawing as is on the study sheet. Like there's pictures in the study sheet, but it's, yes, it is, I promise. Study sheet, what I mean is the study sheet, those pictures that come along with the lab manual. Like the study guide, what's it called? Lab final study sheet. For lab, it's in the lab manual, so I know for a fact that you have it. Not a drawing, it's pictures, right? It's not got a chart, it's just pictures of tubes, yeah? On that. So you guys make your own drawing from that, right?
Yeah.
Would you learn better from doing it yourself? You don't like it. You don't like it. So I'll do you this. I think I uploaded the slides for the lab. Remember when I did lab midterm and somebody asked me to upload the slides? It would be an old announcement. Let me see if I can find it for you guys so that you guys don't have to look for it. Let's do it together. You'll have it. I promise it's in the announcements. If you want the actual drawing thing of it. Let's do it. Yeah, because it was early on, because I remember, I think it was you, maybe. Somebody asked me for the slides, yeah. It's worth looking for it. We'll find it together so you guys can find it easily. Just wait for me to do my duo push. $803. Okay. Get hot in here. I'm moving around a lot. So we go to our course. Just go to announcements, especially if you're on your laptop. You can't do this on the phone, right? You can't search the announcements on the phone. But if you're on a computer, you can. Slides maybe. Overhead. Okay. Lab overhead slides. So yeah, look for overhead if you're searching. Otherwise, I posted this on June 11th. So you can find it by sorting through finding June 11th. Lab overhead slides. And it should have all of the overhead slides on there for the whole semester. So yeah, it's got the midterm stuff, which is probably what we were paying attention to in the beginning. But if you scroll down, you will get to the bottom. And there you go. That help? Yeah. Yes. So there is that. So there is your drawing there. Good question. I'm happy to help you guys find it. That way you won't be searching for it forever. It was June 11th if you're scrolling through all of them. Or you can search on your computers for overhead, and then that'll be there. Or you know, take a picture of this with your phone right now. Also, I don't care. So, but it's here. I'm trying to center it better. So I'm going to zoom out, and then you can see the whole thing. Anyways, so there is that. Good question. Let's get back to the Kahoot. Okay, so back to the thing. Uninoculated. If I tell you this is inoculated and I added A and B only and it turned red. So what were we detecting? Nitrite, right? So was this tube positive for nitrate reduction? Yes. Okay. If I tell you this is uninoculated and this had A and B and then zinc and it turned red, what does the zinc detect? Nitrate. So if zinc detects nitrate and it turned red, it's the ultimate question overall. I might just give you this. I added A and B and zinc. Was this tube positive for nitrate reduction? No. No. Why? Because nitrate is still there. We found nitrate to still be there if it's red, and we added A and B and zinc. Nitrate was detected by zinc Nitrate was not reduced. Remember, if you're reduced, you have changed your chemical compound. You're not going to be nitrate anymore. Okay? If I added A and B, this is uninoculated and this is inoculated. If I added A and B and did not add zinc, A and B but no zinc, and it looks like this, A and B but not zinc. Did I reduce nitrate? You can't tell. So you can't say yes or no. You can't tell until we add zinc, right? All right. If I tell you that I added A and B and zinc, and it looks like this after A and B and zinc, A, B, and zinc, and it looks like this. Did we reduce nitrate? Did we reduce nitrate if it looks like this? Excellent. Is it nitrite? No, it's something else. And you don't have to worry about what. Not nitrate, it's not nitrite, it's something else, but we did reduce nitrate. Okay, are we tracking? I know this is the hard one. This is the one that gets the people. So take your time to go through this one. We have that chart now to refer to that is posted on your announcement to help you guys with that studying, to sort that out. Talk yourself through it, work it out, make sure you understand what those colors mean with what reagents that were added. Okay. Yes. Say, don't forget the gas. It has a Durham tube. You won't have a tube that has gas in it, but if it did, it would be nitrogen gas, N2. All right, next. Which test is this one? Have an uninoculated tube. Here's some examples of some results that will be on the test. See this one that's uninoculated result. All right, so this is TSIA. How do we know that it's TSIA? Because we've got all these colors of anywhere from yellow to pink, and we had some tubes that had the black precipitate in it. TSIA, triple sugar iron assay. Some questions about this. Which tube in those images up there show evidence of H2S production? H2S is the product of sulfur reduction. Triple sugar iron assay does test for sulfur production, for sulfur reduction. All right, you make H2S as a byproduct of sulfur reduction. The iron in triple sugar iron assay makes a black precipitate that will be visible. And that would be tubes 3 and 5 have the black precipitate here.
Is PSIA this one?
Yes, yes. All right, multi-select. Which tubes show evidence of gas production? We'll go over that here in just a second too. Which of these two tubes, not including H2S, show gas production? And here is a tube that I will be using on the test that has gas production. All right, so we can see here, there's a pocket of air under here. There's also a bubble on the side of the media, evidence of gas production. I didn't want to use one that was crammed up. That was from table 2. That went real all out with this gas production because it went a little bit, wild. But a little bit of gas production here in this tube, right? Not related to that. So this would be in these images, tube 4 and 4A. They got lifted up by that gas production. This one still counts as gas production, it's just not as much. All right, so TSIA, this is our uninoculated tube. How did we inoculate this one? We stabbed it with the needle and did the slant streak. So be sure that whatever choice you pick, if I ask you how we inoculate it, it has both of those things with it. Stab and streak the slant. How do we read it? This is phenol rad based pH indicator. So we will look for the pH change based on the color. Yellow is acid and pink is alkaline. If you have acid, it means you fermented a sugar. If you have alkaline, it means that you have ammonia from breaking down your nitrogen source, just like our pH scale taught us. In this case, if it's in the butt, that'll be glucose. If it's on the slant, it's lactose and or sucrose. We can't really differentiate between the two. Slant, lactose or sucrose, butt, glucose. So let's look at this. Did this organism ferment glucose? Did it? Did this organism ferment glucose? Yes. How do we know? The butt is yellow. Acid is from fermenting. The butt means glucose. Okay? Acid from fermenting, the butt is glucose. Did this organism ferment lactose and or sucrose? Yeah, it did too, because the slant is yellow. Did this organism ferment sucrose and or lactose? Sucrose or lactose? Did this organism ferment sucrose or lactose? No, right? Because the slant is pink, alkaline. It somehow aerobically used those and made alkaline products for breaking down that those sugars. But we broke down the nitrogen source to make ammonia up there. The butt down here, we can start to see the butt is yellow. It's hard to see it because the black stuff is there, but I promise you it is yellow. This organism did ferment glucose. It's hard to see. I won't ask you about that potentially on that particular tube. They're all basically all yellow. So, you know, those are the tubes that you'll see. They either look like this, all yellow, glucose and lactose and or sucrose fermentation. We made acid and we made gas in this one. The other possible tube you will see will be this one. Alkaline slant, and we've got black precipitate in here. Okay. All right, so those black precipitate for sulfur reduction. Pink means you broke down the nitrogen source, which is peptone, by the way, to make ammonia, which is an alkaline product, changing that phenol red fermentation to pink, phenol red pH indicator to pink. And yellow indicates fermentation. If it's in the butt, fermenting glucose. If it's in the slant, fermenting glucose, sucrose or lactose. Yes. 3 sugars were glucose, that's the butt, and sucrose or lactose, that's the slant. All right, we made it. Through TSIA. TSIA is a lot. All right, which test is this one? It's a broth. All right, so this is the urease test. Beautiful. What's the pH indicator in the urease test? Phenol red. Why is this tube pink? It's positive for urease because the urease broke down the urea into ammonia, which is alkaline, and that turned the phenol red pink. Very pink. These are the tubes that will be on the test. If I ask you which tube was positive for the assay, even if I don't tell you what the assay is, you should know that it's the pink one. Yes? Okay. Cool. You guys are doing great. Which test is this one? We have a slant with bubbles in it. Another one I don't have today because I have to, treat it on the day. We have a slant on which we grew some stuff. We added a reagent and we made bubbles. Everybody always confuses the catalase and the oxidase test. And what did I tell you about confusing the catalase and the oxidase test? So for sure for the exam, be aware, if it's got a liquid in it and it's a tube with a liquid in it, then that's the catalase test. If the liquid is bubbly, then it is positive for catalase. If the liquid is not bubbly, then it is negative for catalase. All right, if it's a swab, the only one that we do on the swab is the oxidase test, right? Be the next question, I'm pretty sure. Yes. Which test is this one? Now there's no excuses. This is the oxidase test. Remember that the reagent that we're using, and I won't test you on what the reagent is looking for the presence of cytochrome C oxidase, which is one of our cytochromes in the aerobic electron transport chain. It's a test for aerobes. So oxidase, it turns that purplish blue color if oxidase is present. All right, other tests that weren't on this, the malonate test. I've showed you guys this one earlier. If I show you a tube that's liquid and it is blue or green, either of these tubes, if I just show you the tube, you guys should be able to tell me that it's the malonate test. It's a broth, it's blue or green. There's only one test that looks like that. It's the malonate test. If it's blue, this is the pH indicator bromothymol blue. Blue means alkaline was made because we broke down malonate and were able to use the nitrogen source and make ammonia, which is alkaline. All right. Phenylalanine deaminase. It was a yellow slant, little top slant. I'm not going to test you that it's a yellow top. Don't worry about the top colors. I'm not going to test you on that. Okay. But it's a slant that we would add ferric chloride to. If the slant turned green, then you were an organism that had the enzyme phenylalanine deaminase, meaning you removed an NH2 group from the amino acid phenylalanine. Ferric chloride, FeCl3, is the reagent that we add to phenylalanine deaminase. Turns the slant green. MRVP, we had a clearish broth. It just looked like a broth. It looked like this, basically. It was a broth. And then we split it into those tiny tubes. We added either methyl red to test for heterolactic fermentation, or VP to test the VP reagents, which were 1-naphthol and potassium hydroxide. Let it sit for 30 minutes. And that was the Vogue's ProScour test, testing for mixed acid fermentation. They are not pH dependent. They are basically chemical component dependent. And essentially, the methyl red will be red is positive for heterolactic fermentation. Orange is negative. For the VP test, a red layer at the top meant that you were positive for mixed acid fermentation. And if it stayed kind of a golden brown, then it was negative. All right, the gelatinase, most of us are familiar with this because most of us did use this one as our example. And of course, both of mine are liquid today. So I will try to pick a tube that isn't. Let's pretend. Okay. So, right. So if I tell you this is uninoculated and this one's inoculated and it is liquid here at room temperature, this organism did produce gelatinase and it liquefied this media. And I'll be sure that at room temperature that it is behaving the way that it's supposed to for you guys. Because I probably wouldn't put gelatinase it holds its shape. But anyway, so liquid will be gelatinase that breaks down the actual gelatin, which is our solidifying component in that gelatin media. How do we inoculate the gelatin media? Dab with a needle. All right, let's get back to our basics really quickly. How would you classify this organism's characteristic overall arrangement? The one with the arrows, one I want you guys focusing on. Good example. Almost done. All right, that's strepto. That is chain. Yeah, I don't know what else to say about it. Strepto's the chain. All right, remember Staphylo is irregular bunching. Those are the only two you'll see on the test. What is the name, the name of the shape of these bacterial cells? The name of the shape. A shape, not the arrangement. Shape of the cell. All right, these are bacilli. These are rod-shaped cells. Excellent. So you will either see bacilli or cocci. Those are the only shapes that I have. You will either see strepto or staphylo. Those are the only arrangements I have. So just focus on those ones and you should be golden. All right. Shapes, like I said, bacilli or cocci. Bacilli are rods, cocci are round. Arrangement. Staphylo is the irregular grape-like clustering. Strepto is the chain. Don't get shapes or arrangements confused. If I'm asking about the shape, tell me about the shape. If I'm asking about the arrangement, tell me about the arrangement. Remember that simple stains are a thing, right? That if we do a simple stain, it just helps you look at contrast. It doesn't tell you whether you're gram positive or gram negative. If you are gram positive and we perform a simple stain with methylene blue, what color will you be? Blue. Blue, right? Methylene blue. It's just blue. It don't matter if you're gram positive, gram negative, acid fast, eukaryote, a fungi. It doesn't matter. If we do methylene blue simple stain, you blue. Right? Don't get tripped up on those details. All right, microscope use and parts. If you observe a specimen at 100x objective lens through a 10x ocular lens, what is the total magnification? $1,000. So be sure you're reading your numbers and your zeros. So you just add 1 0 onto the 100. That's 1,000. All right, which lens is used with oil immersion in our microscopes? Right, that 100x. We don't even have a 500x lens. Know your parts of the microscope and what they do, what their functions are. Don't be tripping up on that one. Lab safety, all those stains that we reviewed, the differential and selective medias, the biochemical tests that we went over, and then be able to read an identification chart and use a dichotomous key. So what do I mean by that? What I mean is, if I give you a form that was like this one, the one that you guys turned in, that first page, if I give you a form completely filled out, and I give you a dichotomous key, I will give you both, okay? But if I give you this, and I give you a dichotomous key, like this one, you should be able to tell me what the organism is. If you're not confident in that, practice. If you really can't figure it out, ask me and I'll help you. So I walked you through it in class. I'll be able to, you're always going to start on the left and work right. Always ask the questions in order. Always in order. Do not skip around when you do those, okay? There will be 2 questions, 49 and 50. On one of them, I will give you a chart, like the ones that we filled out like this, and a dichotomous key. And I will ask you, what is the gram-positive organism? On the other one, I will list out test results. I'll just give you a whole big paragraph with test results. I'll just randomly, it was positive for urease. It was, it reduced sulfur. It did these things, or it was negative for these tests. And I will give you a chart. And you'll have to tell me what the organism is. We'll do poorly on those questions typically, which I feel like are gimme questions, you guys. That's start with the left, ask the first question. Based on that result, you stay where you are and keep asking the next question. Do not deviate from your route on your chart. Do not deviate. If their question is gram positive and the next question is, was the bacilli or a coccus? If it was a coccus, the next question is, produces catalase or not. Don't look on your thing if it has nothing to do with the cocci. If yours is bacilli, don't be looking for catalase, for example. Okay? Don't get caught. Follow the track on your key. Your key is what you're referring to more than anything. Both your chart or that paragraph that I give you of all those things are going to have way extra information that you might not need. Track what you need to, following the dichotomous key. Is everybody good? Okay. All right, that's it. Good luck studying, everybody. Here's our final podium. And if you guys, when you're studying, you can play this on your own. You guys know you can play through this on your own. And we also have the lab Kahoots that I posted in a previous post to help you guys with studying. And if you guys have any questions, please email me and let me know. The sooner the better. Good luck, everybody. And I'll see you tomorrow for the exam. Lab final tomorrow. Thursday.
Said the questions, like, unit 4 is gonna be like unit 4 questions, but everything else from like unit 1 through 3 is gonna be like can't answer them unless you know other things. But
It's kind of a mixture, like some of the unit 1 through 3 questions will be just straightforward questions. But some of them will be unit 4 plus. Unit 1 through 3. Yeah, and there will be unit 4 questions that you will have to know something from unit 1 or whatever. So I'd say it's a mixture and match. Some of the unit 4 questions will be just unit 4. There will be critical thinking questions that are based on unit 3 as well. Yeah. There'll be critical thinking questions in 1 through 3. Yeah. So yeah, just think of it as a mixture of all the 4 units all at the same time and sit that way and be prepared for that. One of the common things I often ask on unit 4 questions will be, was this a bacteria, virus, protozoa, or helminth? That's a very common one. So it'd be like, your patient is diagnosed with tuberculosis. Which type of pathogen caused this? And it might be bacteria, protozoa, blah, 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 blah. I also might ask the same question. Literally, I might have the same question, but instead of asking what type of organism, what is the name of the species? You know, and so you have to not only know that it's Mycobacterium tuberculosis, but that is a bacteria. Luckily, Mycobacterium tuberculosis. But you know what I mean.
Yeah.
So malaria, Plasmodium, that's a protozoa. And so that's pretty common one. Having to know that it's virus, protozoa, helminth, and whatever, and knowing the organism name associated with those diseases. So that disease compendium chart that I have, go through those. If it's not on my lecture, like learning objectives list, if it's not on there, then don't study it. Like just take it out of the list. You can delete it in like the Excel file, like when you guys are studying. The Excel file for that disease compendium thing also has, let me actually pull it up so I can show you. You guys are studying for this, because we might go over this too, for the actual final, like lecture final stuff. Is super slow. I'm gonna feeling that it's gonna be that way whenever we take the test too. I think it's neat. So anyways, if you actually download this and open it in Excel, which is what I would recommend doing. It's opening, it's just, oh, it's on my other screen. I'm like, where is it? Right here it is. So over here, type of organism. P is protozoa, V is virus, H is helminth, B is bacteria, F is fungus.
Okay.
Okay, and so you can actually sort it by that. So if you go up to here, enable editing, you'll have to enable editing, FYI. And it's going to come over back to my other screen. Of course it is. Okay. So after you enable editing, it'll let you do that. Better, okay. You can sort it. And you can sort A to Z. So now these are all the bacteria, all the fungi, all the helminths, all the protozoa, all the viruses, and there's some prions hanging out in here. So you can sort it that way. You can sort these alphabetically by the species name. You can, if you just want to see bacteria, you will uncheck that and then just check the bacteria, and it's just the bacteria.
And what about the symptoms of those? We need to, like,
Some of them it's important, right? So what I'll say is, you don't need to sort them by that probably, right? But I think it's up here in case you did want to see them. But like, for example, if we have associated diseases over here, there's some where we can actually look at this. F. I clicked Alt, that's why. I want to open it in that sheet. It's not letting me do it. Okay. Okay, so you can search for meningitis. You can also like look to the associated diseases here. Find meningitis. Right here. Here we go. So we have meningitis down here, several causes of it. We have fungal, bacterial, and whatever. And you can search for it and find meningitis on here, and then search for them through here as well. Okay,
okay.
so they can find that anyway. So if we look at meningitis here. Meningitis, headache, photophobia, petechiae, rash. Neisseria meningitis has the rash associated with it. But you notice here, signs and symptoms for this one just says meningitis. So I would assume you would know meningitis causes headache and photophobia, but the rash is particular to Neisseria meningitis. None of the other ones cause a rash, typically. And so that's a characteristic of that. So I would know signs and symptoms that are important for like unique diseases, for like syndromes. So I would know that meningitis has, headache and stiff neck and photophobia. And fever, but you don't need to know literally every single detail of every single science, and just where it's important for differentiating maybe. So if I tell you that, your patient has things that tend to you that's meningitis, but that they have a rash, then you should know that it's probably Neisseria meningitis and not Streptococcus pneumoniae that caused it. You know what I mean? So using you guys, so I know when you guys are making your concept maps, you guys should put them together thoughtfully, but you're knocking it out at the same time. Make sure that you're going through your concept maps and making sure that you're really like identifying those differences between that stuff. Because like that's where that will be useful for studying this thing.
Okay.
Other questions?
No.
Okay. Good. All right. Yeah. Thanks for coming, guys. I really appreciate it. Get out of it.
I just didn't know if you thought I needed to work first tomorrow.
Okay, go ahead and wear them. Okay, just for your own protection. Yeah.
I just didn't know if you weren't like opening the things and stuff.
Yeah, just in case some people might spill them, and just in case somebody spilled it and you pick it up, that way you're protected. Okay. Yeah. Yeah.
Thank you.
Yeah. All right, if you guys are joining online, that is it for today. Thank you guys for joining. I will post the thing as well.